Mitophagy, the specialized form of autophagy responsible for selectively degrading damaged or dysfunctional mitochondria, is increasingly recognized as a critical safeguard of cellular health. Mitochondria, often described as the powerhouses of the cell, are essential for energy production, but they are also major sources of reactive oxygen species, RLS, and pro-apoptotic signals when damaged. Left unchecked, defective mitochondria not only impair cellular metabolism but also promote genomic instability and inflammation, two conditions that are tightly linked to both aging and cancer development. Through mitophagy, cells can remove these malfunctioning organelles before they cause systemic harm, making this process a linchpin in the defense against degenerative disease and tumorigenesis. With age, however, mitophagy becomes progressively impaired. Key regulators of mitophagy such as PINK1, PTEN-induced kinase 1, and PARKIN, which tag damaged mitochondria for degradation, become less responsive or are epigenetically downregulated. As a result, older cells accumulate dysfunctional mitochondria that produce excessive ROS, triggering oxidative damage to nuclear DNA and telomeres. This contributes to cellular senescence and chronic inflammation, hallmarks of aging, and sets the stage for age-related diseases, including cancer. In this context, mitophagy functions not merely as a cleanup mechanism, but as a vital line of genomic defense. Its failure allows the slow buildup of mutagenic stress that accelerates both tissue degeneration and malignant transformation. In cancer, the role of mitophagy is highly context-dependent and complex. In early tumor suppression, mitophagy is beneficial. It clears mitochondria that might otherwise generate oncogenic mutations or metabolic imbalances. But in established cancers, especially those in hostile environments like low oxygen or limited nutrients, mitophagy can support survival. Tumor cells may exploit mitophagy to maintain metabolic flexibility and resist apoptosis by removing mitochondria that could trigger intrinsic cell death pathways. This paradox, where the same process that protects against cancer initiation later supports tumor progression, underscores the difficulty in targeting mitophagy therapeutically without causing collateral harm to healthy tissues. Despite this duality, the therapeutic modulation of mitophagy holds promise. Agents that restore or enhance mitophagy in aging tissues could improve mitochondrial quality control, reduce oxidative stress, and delay both functional decline and oncogenic mutations. Some natural interventions like caloric restriction, intermittent fasting, and certain polyphenol, e.g., resveratrol, appear to boost mitophagy, contributing to their observed effects on longevity and reduced cancer incidence in animal models. On the pharmacologic side, molecules like urolithin A are being studied for their capacity to selectively induce mitophagy in aged muscle, with potential implications for broader anti-aging and anti-cancer strategies. Yet the risks must be carefully managed. Inhibiting mitophagy in established tumors to increase their susceptibility to metabolic stress or chemotherapy is an active area of research. However, this approach must be tightly controlled, as impaired mitophagy in normal cells may accelerate aging and increase cancer risk in the long term. Conversely, boosting mitophagy indiscriminately could inadvertently support tumor survival or drug resistance if applied at the wrong stage of disease. Ultimately, mitophagy sits at the intersection of two fundamental processes: mitochondrial quality control and cellular fate determination. Its decline with age represents a central vulnerability, one that undermines energy production, increases mutagenic potential, and opens the door to age-associated malignancies. Yet when functioning properly, mitophagy is a powerful suppressor of both aging and cancer. The challenge is to restore and target it precisely, amplifying its benefits where protective and restraining its activity where co-opted by disease. In doing so, mitophagy may become not only a biomarker of cellular resilience, but a focal point for therapies aimed at prolonging health span and preventing the slow transformation of time into disease.